Hi, this is Muthukumar, Associate Professor of Pharmaceutics, belongs to SNS College of Pharmacy and Health Sciences. Today we are going to discuss about the sterilization evaluation. So, what about the sterilization efficiency? How to evaluate the sterilization efficiency? So, first, sterilization, you know what is sterilization? It ensures or makes the pharmaceutical products free from the complete viable microorganisms, because it is very important to ensure the free from viable microorganisms. Next, what are the efficiency evaluation is essential to confirm. So what are the evaluation is essential to confirm the products? First, consistency. So without any lagging, it has to be consistently give a proper microbial free products. Next, safety and regulatory compliance. So you know what is safety and regulatory. It will be vary from one country to another country. So based on the country, there must be the adherence towards the compliances. Next, methods. What are the methods going to be evaluated? Moist heat, dry heat, filtration, radiation, gas sterilization. These are the whole methods performed for sterilization. These methods has to be properly evaluated and documented about their efficiency. Next, what is efficiency? It is depends on the microbial load or the microbial accumulation. Next, product compatibility. Indirectly, it's nothing but a product compatibility. And the main thing, another area towards the equipment area, it's a equipment performance, how it performs, how it gives a proper sterilization. Next, sterility assurance level, that's nothing but a SAL. What about the sterility assurance? Whether it gives the proper assurance towards the sterility mean by the regulatory complaints. Next, what is the goal? So the main goal of the sterilization efficiency is nothing but achieve sterility assurance level SAL 10 to the power of minus 6, meaning less than or equal to 1 non-sterile unit per million. Next, what are the key parameters for evaluating sterilization efficiency? First, biological indicators. Simply we call it as BLS. So what are the biological indicators? Geobacillus stereothermophilus and Bacillus atrophius. So they have capability to withstand the high level temperatures. That is the reason why here we mention those three microbes comes under the category of the biological indicators, because they contain highly resistant spores. To kill the spores is not at all so easy. So these two microbes used to validate steam, dry heat and gas sterilization. Next, chemical indicators. What are the chemical indicators? There is a color changing labels indicating exposure to sterilization conditions. There are so many color changing chemicals attached with the sensors nowadays. So they fitted inside the cabinets and it is going to evaluate the sterilization efficiency, like how the conventional phenolphthalein indicator changes its color to give the result. The same thing only here. Once the efficiency of the sterilization is over, based on the efficiency, it develops the colors. Next, of course, we know the physical parameters: temperature, pressure, humidity, and radiation dose. It's only for radiation area. Next, gas concentration, for example, ethylene oxide, and exposure time. How long those things are exposure towards the sterilization process. Next, of course, to identify the SAL calculation. The next important thing is D value and Z value. So D value is nothing but the time required to reduce the microbial content by 90% of its total. Z value is nothing but it's a temperature change. These temperature change needed to change the D value. It's nothing but reduce the microbial population by 90% by a factor of 10. Next, evolution of the moist heat and dry heat sterilization. So we know the temperature range of the moist heat as well as the dry heat sterilization. So here the moist heat, the temperature range is 121 degrees Celsius to 135 degrees Celsius, and the dry heat sterilization 160 degree Celsius to 180 degree Celsius. So what are the indicators? So for the indicators of the moist heat, example autoclaving, of course the biological indicators in previous slide what we discussed, both we can mention. Here we mention. So geostereothermophilus spores. Next, it monitors the temperature, pressure and valve category. Next, the important test is Bowie-Dick test for the steam penetration. So, Bowie test is nothing but the identification of the steam penetration. Next, indicators for the diheat sterilization. So, we can give the Stereothermophilus as well as the Atrophius bacillus species. So, both are advisable for the dry heat as well as the moist heat sterilization. Next, physical monitoring of the temperature uniformity, how it uniformly spreads the temperature inside the cabin. Next, verification of the oxidative pyrolysis and T-biogenation. It's a special, it doesn't perform under the moist heat, it's exclusive performance for the dry heat sterilization. Next, evaluation of filtration, radiation and gas sterilization. So filtration, so the sterilization efficiency evaluated by the integrated testing is nothing but a bubble point, diffusion test and pressure hold test. So microbial retention validation using the microbes driven demonas and diminuta. It's a specialized microbes. When compared to the other microbes, it's a so small in size. That is the reason why here we take these microbes as a sterilization efficiency evaluation. It is the smallest known bacteria at present. Next, radiation sterilization. We know the example gamma and electronic beam. So, dose measured is nothing but a kg. We navigate values. What are the evaluation parameter? So dosimetry mapping. Dosimetry mapping is nothing but how much dose we design to give the uniform dose distribution in order to perform the sterilization evaluation. Next, BLS change is a biological indicators challenge with Bacillus pumilus. Here for radiation sterilization, we prefer the Bacillus pumilus microorganisms. Next, gas sterilization. Here the example is ethylene oxide. So what are the evaluation parameters? Gas concentration, humidity, exposure time and temperature. These are the unique parameter comes under the category of the evaluation or efficiency of the gas sterilization systems. Here the biological indicator is nothing but the same Bacillus atrophius. Next, residual ethylene oxide level. It must meet safety limits as per the ISO requirements. Next, final assessment, validation and documentation in the industry side. So what are the final validation and documentation we are going to see? Sterilization efficiency, it must undergo the category of the installation qualification, operational qualification and performance qualification in industry. Especially sterility testing. After sterility testing over, it confirms the final product sterility using membrane filtration or direct inoculation. These are the two indirect tests to evaluate the product. After undergoing sterilization, the product sample has to be taken and it go for the two methods of the sterility testing as per the USP. The first category is nothing but the membrane filtration and the second category is the direct inoculation. Next, revalidation. It occurred periodically or after the equipment modification. So, in between there is a change of the equipment or operational qualifications, whatever may be. Again, it has to be rechecked and revalidated. Next, the all documents, all results making a proper documentation as per the GMP and regulatory guidelines. Of course, already we discussed it will be vary from one country to another country. Next, what is the main purpose of the sterilization evaluation? So it gives a consistent and ensure the consistency of the production of pharmaceuticals pipeline. Next, reproducible sterilization, whether it is need a reproducible sterilization or not. Next, so what we discussed, ensure of the consistent and reproducible category of the sterilization. It has to be go uniformly across through the all pharmaceutical batches without any variation. So for that reason, the assessment of validation and documentation under the sterilization evaluation must be very important.